Hi, everyone, and welcome to episode 127 of Silencing the Podcast. I'm Erin Harris, and for this episode, I'm joined by Carlos Buesa, Ph.D., founder and CEO of Oryzon, which is a pioneering epigenetics company that he launched back in 2000. Carlos has a Ph.D. in biology, a background spanning the University of Barcelona, the Flemish Institute of Biotechnology, and leadership roles across Europe's biotech ecosystem. And so he brings both scientific depth and strategic perspective to our conversation. He and I talk through how his team's early work in functional genomics led them to LSD1, an epigenetic target now showing promise across oncology, CNS disorders, and even sickle cell disease. We get into everything from advances in chromatin biology to new clinical data in AML, along with what it takes to turn epigenetics into real-world therapies. It's a great look at how precision approaches like LSD1 inhibition could start to reshape treatment across multiple diseases. All right, well, I'm going to let you jump into my conversation with Oryzon's Carlos Buesa. Enjoy. Carlos, hi, welcome to Silencing the Podcast. I'm so happy you're here.
Thank you very much, Erin. Very pleased to be here.
Wonderful. So talk to us a little bit about Oryzon. Tell us a little bit about the company and its mission.
Well, the company is a publicly listed company in Spain, in Europe, very liquid, funded by a couple of scientists 26 years ago. At those times, we were just a niche platform, functional genomics company. Well, over time, we evolved to create our own molecules. And in the last 12, 15 years, we have been becoming specialists in epigenetic drugs. And our mission from the starter, was exactly to try to identify what genes are responsible for diseases, and later on, just to develop drugs that could help patients. So this is our mission, our passion, and is what we are committed to.
Excellent. Okay, so I want to talk a little bit about how you have been a pioneer in targeting LSD1 for years. So I guess for listeners who may not be totally aware of LSD1, tell us a little bit about that. And then how has the scientific understanding of epigenetics evolved, and what gives you the confidence that LSD1 inhibition can truly reshape treatment paradigms in both CNS and oncology?
Yeah, well, it's a very long story. We end up in LSD1 because we were doing some genomic research on post-mortem brains from patients that had died with initial symptoms of neurodegenerative disorders. So anyway, we end up in LSD1. LSD1 was at that time an obscure target recently described by the Harvard University team. And then we and others, we were learning about the biology of LSD1. LSD1 has a sort of dual life. On one hand, is an enzyme. As an enzyme, what it does is demethylating a number of proteins, mainly the lysine residues of histone proteins, and most concretely in histone 3. And by doing that, is basically contributing and changing the morphology of the chromatin and opening or closing, depending on the context, the folding of the chromatin, allowing the transcription machinery to start doing this job. But also, it's also the methylating other enzymes which are important, like, for instance, P53. Some of the data that we are having right now might be explained also by this additional function. But as I said, this has two main roles. This is a kind of Dr. Jekyll and Mr. Hyde enzyme, because the other role is absolutely independent of the enzymatic role, and is a scaffolding role. LSD1 is a key element on a number of transcriptional complexes assembles that are playing a fundamental control in the promoter and enhancer and super enhancer regions of specific areas of the chromatin and from important genes. So this is basically what the enzyme does. Over the years, we and the rest of the community, of course, we have been basically deploying efforts to understand the fundamental biology. And now we know that it plays a gigantic role in CNS and in oncology. So epigenetics was for a time a sort of area of big hope when the ACE/DAC inhibitors were discovered and developed, and then very soon it came to fact that ACE/DAC is a very complex superfamily of deacetylated and deacetylase proteins, and it was difficult to get sufficiently selective ACE/DAC inhibitors to get the real therapeutic activity without side effects. But the second generation of epigenetic agents that were discovered, LSD1 as one of the histone demethylases, but also histone methyltransferases and readers like bromodomains, were offering a second opportunity, a second chance to the field to develop highly selective therapies. And LSD1, I think, is a beautiful example of that. So right now we have a strong confidence that LSD1 can reshape treatment paradigms in oncology and in CNS. So in oncology, we have seen that LSD1 plays a fundamental role in interacting with the gene expression of the cancer cells to make them visible to the immune system. It's also interacting with the gene expression of the T cells to make them more efficient to capture the cancer cells. And it's also make them more resilient and therefore, less prone to become exhausted. In certain myeloproliferative disorders, like leukemia, we have seen that LSD1 is key for the maintenance of the leukemic stem cell. So these repositories of these small repositories of hard to kill less differentiated cells, which are at the end of the day the ones that are going to be responsible for the relapse, are basically needing LSD1. So when we are inhibiting LSD1, we are doing a sort of synthetic lethality. And that's only for oncology. And of course, it's not a silver bullet. It's only working in a certain subsets of cancers. But we know that we can do a surgical activity on those specific cancers. In CNS, the story is also equally fascinating. We know now that epigenomic dysregulation is a core feature on neurodegenerative and neuropsychiatric disorders. It's very interesting to note that a significant proportion of the novel mutations are basically associated with a autistic spectrum disorder, with schizophrenia, and other psychiatric disorders. And we have seen, actually, in post-mortem brain of autistic patients that they have a diminished demethylation in the lysine 4 on histone 3, precisely where we are having an activity in prefrontal cortex. And also these patients saw a general decrease on certain of the expression of certain epigenetic genes, which are important. And thus, it's a convergent evidence with the fact that when we and others, we treat mice which are representing models of some monogenic variants of autism, LSD1 inhibition can compensate the phenotype, and that has been demonstrated up to now in 6 monogenic CNS ASD-like disorders. So this rescue, which can be partial or almost total, is giving you an evidence that basically rebalancing the methylation status of the chromatin in the prefrontal cortex and in other areas of the brain might be key to basically recover some of these functional impairments.
Terrific. Okay. I want to talk a little bit about the ALICE-2 trial in acute myeloid leukemia, or AML. And we know that it has shown striking early efficacy signals. So why do you believe, or what do you believe, such high response rates with yademstat, in combination theory. How are you thinking about differentiation in the increasingly competitive AML landscape? Where are we?
Well, it comes again to the mechanism of action. I think that what is clear is that Yadadenstat, LSD1 inhibition with our molecule has a dual role on the leukemia. On one hand, we are having this scaffolding activity that I mentioned. We are disrupting our compound is basically binding covalently to the catalytic site of the enzyme, and there is a protrusion of the molecule which is producing an impairment to LSD1 to land on the protein-protein interaction with GFI1. This is basically the repressor complex, which is impairing the normal differentiation of leukemia cells. We are basically disrupting this, and by disrupting this, we are forcing the cells to go to a more differentiated status where they can immediately enter on apoptosis. So we are contributing to the pro-apoptotic mechanism, but the other part of the effect is, as I said, is the leukemic stem cells. So we are hammering the leukemic stem cells in a way impairing the replenishing of the leukemia cells. So when you think the current backbone, Ben-ASA, azacitidine is basically an anti-proliferative agent, is basically a sort of chemotherapy. And then venetoclax is anti-BCL2. I mean, it's basically a pro-apoptotic agent. So we are reinforcing the apoptotic arm of this in the triplet, and we are having an additional new part, which is something that other combinations do not apport. So we think that we are very competitive. We think that we are certainly equally or more competitive than the effort that others are doing with other triplets. So we know that many inhibitors, for instance, are right now under the radar because, on the radar screen because they are offering hope. But we think that we are offering differential aspects that make our combination better than this one. The first one is a better potential biological coverage. I mean, of course, many inhibitors are designed for people who have a specific genetic background. I mean, patients who have an AML with an NPM1 mutation. Same thing for the triplets with gilteritinib, which is a FLT3 inhibitor. So in both cases, the proportion of patients of AML having these particular mutations are around 30%. So we know that YADA is mutation agnostic. So we can virtually give this combination, this triple combination, to any AML patient. So this is the first competitive advantage. Second is that, of course, we do not any additional toxicity to the Ben-ASA backbone. So it looks like the triplet is very, very well tolerated, or well, at least as well tolerated as the backbone Ben-ASA only. And in terms of efficacy, we are having not only 100% of responses, but we believe that we have better performance than, for instance, many inhibitors. We have 90% of CRs, pure CRs. And we are going to present it at EHA in June in Stockholm. I think that the data that we are going to present there with 17, 18 patients are going to support that this expectation of being better than many inhibitors is going to be confirmed.
Terrific. Okay. Now, I want to talk a little bit about how this is impacting sickle cell disease, or SCD. So you're also advancing Yadademstat into sickle cell disease. And what is the underlying biological rationale there for targeting SCD with LSD1 inhibition? And from your perspective, what does success look like there?
Okay, no, perfect. This is basically we go to the fundamental the fundament of the disease. What we know in sickle cell disease that there is a mutation on the beta globin. So It is well known that an increase of fetal hemoglobin expression can mitigate or rescue the phenotype of the disease. And we have a beautiful example in the Persian Gulf, not because of the reasons that we have right now, but because of human genetics. There is a population in Saudi Arabia that they are basically carriers of the mutation in the beta globin gene, and yet they are not developing the disease. And this is because they have other mutations that they do basically like a suppression genetic effect, that these other mutations produce an overexpression of the fetal hemoglobin gene. So this is a wonderful human fundament of why this approach is a very meaningful one. In fact, FDA approved Casgevy, which is a gene therapy which is based on knocking down BCL11A, which is a core element of the transcriptional complex that repress the expression of fetal hemoglobin. But guess what? In this transcriptional complex, there is also LSD1. And if we repress LSD1, if we inhibit LSD1, we see that there is, we see that we can basically decouple this complex and again overexpress fetal hemoglobin. And of course, it's a much more suitable and affordable approach instead of preparing a patient for a very painful bone marrow transplantation, very expensive, $2 million per patient, to a single pill per day. So I think that the approach is clearly more compelling. We have very strong preclinical data in monkeys, also of course in rodents, but also in monkeys in single shot, in long-term treatments, 10 months. So we are basically having a very strong preclinical conviction that the mechanism works. Right now we are starting this clinical trial here in Europe, Restore, where we have already those 2 cohorts. And for us would be success would be to be able that we are able to increase the fetal hemoglobin to margins which are sufficiently high to rescue the phenotype of the sickle cell disease, so over 20%, without any additional toxicity.
Gotcha. Okay. I want to address perhaps for our corporate strategy minded listeners, who are thinking about novel mechanisms like LSD1 inhibition that a market can be pretty cautious about. So how do you translate cutting edge epigenetic science into a compelling value story for investors?
Well, there are trends and which can be favorable, and trends that can you can be a bit stained. But the question is epigenetics is like other areas of the biology, like phosphorylation, protein kinases, and receptors, and something like that. Biology is a choreography of multiple mechanisms of regulation. And epigenetics is a mechanism that in general we are understanding now better and better, the fundamental biology, the interaction with other mechanisms, with signaling pathways, and so on and so forth. At the end of the day, this is an industry which is data-driven. And when we are presenting data to investors or to other companies showing that we have 100% of responses in a clinical trial in leukemia, in AML, in newly diagnosed patients, this is something that immediately, raise attention. So I am not particularly concerned about epigenetics not raising the necessary attention. For us, like for many other small companies, we are here playing with the savings of lots of small institutions in Europe, small investors, and also more big funds. So we are very aware that this is something that we have to treat with utter respect. And although this is a very difficult industry with a high attrition rate, what we believe is that so far the ability that we have had to navigate around this central target, which is LSD1, we think that we are one of the teams in the world that knows better the biology of LSD1, allows us to get the necessary cautions and keys to play the best clinical development in oncology and in CNS. So we hope that we will be able soon to get to the finish line in AML and in CNS.
Terrific. Okay. Now, before I let you go, let's talk a little bit about Oryzon's roadmap. And I guess my first question is, do you intend to remain focused on your epigenetics platform, or do you see yourself evolving into a multi-asset commercial organization? And just kind of talk us through maybe what the next 2 to 5 years look like.
Yeah, no, that's a terrific question. I mean, we have been surviving over many crises and nuclear winters in the sector because we have been extraordinarily flexible and being able to navigate the circumstances. Now we are in a specific circumstance where we have amazing data in oncology, and that could be the basis for us to commercialize our own products on certain specific markets. At that moment, if we become moving forward a company which is having a commercialization effort, then probably other aspects may play a role, like what kind of assets could be meaningful to complement the franchise, and so on and so forth. We are not yet. We are not yet in this point. We didn't arrive to this bridge. We will see. It is something that certainly what we are really committed is to get the drugs as soon as possible to the patients. If that means that we are going to commercialize the leukemia ones, I mean, that's one thing. Certainly on the CNS, we are involved in bigger indications. I mean, we are working on psychiatric indications with multi-billion market potential. So that requires the effort of a big organization. So there we are more focused on a partnering. So we will see how we adapt the multiple options that we have in front of us.
Terrific. All right. Well, that's a wrap on this episode featuring my guest, Oryzon's Carlos Buesa. Carlos, thank you so much for your time. This was extremely insightful, and we will be following along with Oryzon and perhaps have you back next time this year, next year, this time, to give us a progress update.
Thank you very much, Erin. It has been a pleasure.
And thanks, listeners, for tuning in. Please subscribe to Cell and Gene the podcast. Tell your peers to do the same so you never miss an important episode just like this one. And we'll talk to you soon.